All right, there you go.
All right. Okay, so hi, I'm Peyton. I am a developmental biologist in training. I am still a graduate student, so keep that in mind. And I'm going to start off my talk kind of talking about my past and what I've done to get to where I'm at now, which is in grad school. And then I'll talk to you guys a little bit about what I'm actually doing for my research. So, how fish actually develop their patterns, and then the types of things that I'm doing in the lab with them. So, to get started, a little bit about me. So, I actually grew up close to Orlando, really close to Disney World. So, I spent a lot of time near Disney. Never been to Disneyland though, on the West Coast by where you guys are. So, I'd still like to do that at some point. And I was a cheerleader for 8 years in high school and college. I have a cat named Momo and a dog named Leo. You can see them in this picture here. I was trying to get a good picture of Leo in his new Christmas sweater, but Momo got in the way, and that pretty much sums up uh, their relationship. So, also in my free time, I like to cook and play Nintendo games like Animal Crossing and stuff like that. So, I keep myself busy outside of the lab as well. But for my career path, I really loved science in high school to the point where I took extra science classes my senior year instead of taking like the easier classes, easier classes like the cooking classes and the things like that. So I took anatomy and physics and microbiology and genetics, and I really liked them. So I knew that I was going to be going to college for biology or something in the natural sciences. So I ended up going to the University of North Florida in Jacksonville, where I got my bachelor's of science in molecular and cellular biology. And while I was there, I also worked at the Mayo Clinic, which is a really big research hospital. And I was in a lab that researched Parkinson's disease. So, for anyone that doesn't know about it, it's a neurodegenerative disease that typically affects older people. And it causes basically a loss of movement. So, a lot of shakiness. People have a hard time walking. And it also is connected with dementia and other neurological issues. So, I did some of that for a while and realized I really liked research. I liked being in the lab and doing bench work. So I applied to get my master's degree. And now I'm at Indiana State University up in Terre Haute, Indiana. So now I work and teach and mentor other future scientists like Jackie here, who is one of my students. And I obviously work with fish and colors. So fish are not the only ones that have really pretty, colorful patterns. We see it throughout the animal kingdom. And so we'll get into some examples like we see here. But in my opinion, and maybe it's subjective, I think fish are the best example of it because you see just such a wide variety of different color patterns compared to other animals like mammals like us. So why spend so much time and energy and money studying color patterns, which may seem to you guys like maybe it's not, that important. Like it's just a visual thing, right? So it's just a pretty thing to look at. But color patterns play a role in lots of things that animals care about and a lot of things that are important for evolution. So here's some examples and we'll go through them. So signaling to predators. All of these animals here to the right have something in common. They are all poisonous or venomous. And they all also have really distinct, bright, colorful patterns. So they're not trying to hide themselves away from predators. They're putting themselves out on display and making it obvious that they should stay back, right? Don't attack me because I can bite back and I'm dangerous. And something you'll see a lot also as a side note, is that with these species that are poisonous or venomous, you'll see a lot of blue coloring. And that's actually a pretty rare pigment in nature. You don't see a lot of just natural blue pigments. So I thought that might be a fun fact. And then camouflage is another big thing. So either for prey, so like this Arctic fox here, this is the same species of fox, just one is in the summer on the left when there's no snow, and then the other is in the winter time. So they change their coat color depending on their surroundings. And the zebras also use their pigmentation as a way of camouflage. So they kind of hide themselves next to each other, and so predators can't tell where one zebra stops and the other starts. And so this is kind of a confusion tactic. And it helps keep predators from eating them, unless you have some sort of genetic mutation that affects your pattern, like this zebra on the left. It has a very different pattern, and so it's pretty obvious. It stands out, and so it would probably be more likely to be preyed upon. On the other end of the spectrum, you have fish like this shark that are apex predators, meaning that nothing is really hunting them, but they still want camouflage because they don't want their prey to see them and then run away or swim away in this case. So this shark has a really dark top side and a really light belly, and so it helps it blend in with the water. So if you're on top of the shark looking down, right, you have all the really deep water underneath, and then you see the dark side of the shark, so it blends in. But if you're underneath the shark looking up, you see the white belly, and that makes sense because it's going to blend in more with the shallow water near the sun where it's all light. So you'll see this a lot with shark species where they'll have the two-toned, and that's to camouflage themselves from fish or from their prey. So I have a little exercise for you guys. Can you find the seahorse in this image? If you think you found it, you can maybe go ahead and type in the chat that you found it, but it's hiding somewhere in this coral. Anyone found it yet? Oh, we got a message. Yeah, okay, so hopefully you guys can see my pointer. But the seahorse is right in the middle. You can kind of see its little yellow tail wrapped around the coral. And then here's the stomach, and here is the head. So the seahorse blends in really well with its surroundings. If I can click forward. This is what it looks like close up. And so, yes, a seahorse is a fish, just for those of you that are curious, it is a type of fish. And this particular species is called a pygmy seahorse. They're really small, and you can see that their coloration is almost exact to the type of coral that they live near. So it's a really, really effective camouflage. Another really important role is mate choice. So we see this a lot in birds, also with fish, but most of the examples people think of are in birds. So usually what you see for fish and birds is that the male is going to be more flamboyant and have really bright colors, a lot of patterns going on. And that's so that they can attract a female mate. And the females will tend to be a little bit more subtle, a little bit more colorless, right? More muted tones. And that's because they don't really have to go out of their way to attract a male the same way that the males do. It tends to be female choice. So if anyone's ever owned a betta fish, like this one over here, you probably owned a male betta fish because they tend to sell better at the pet stores. They're a lot more brightly colored, and they have like the big pretty fins, whereas the females tend to be a little bit more dull in color. Also, communication is a big one. So fish obviously are living underwater. They're not really making sounds at each other to communicate. So they have to rely on other methods. And they have chemical methods and electrical methods. But they also have their color patterns. So they can change their appearance to communicate with other fish. So over here, you can see this is a squid. And its skin is just changing color, like just constantly. You can see all of the pigment cells just changing the pigmentation. And so this shows that coloration is a really dynamic process, and it's also really instant. So everything can change about a color pattern really quickly. And that's important because if you're using it for communication and you're saying, hey, look out for that dangerous thing over there, you don't want your message to reach the person in an hour. You want it to get there immediately, right? Time is of the essence. So coloration is a really effective instant method for that. To the left here, this is actually a model species of fish that we use in the lab, in the lab that I'm in quite often. This is the fairy cichlid. And you can see down here this GIF showing this dark patch kind of lightening and darkening. So they change the color of that patch on their face to denote who is more aggressive and who is more submissive within their family groups. So they live in family groups and they raise children together, but they also have individuals that are more dominant and more in charge, which tend to have the really dark black patch there, versus the individuals that are more submissive that have a much lighter patch. Also, in the case of humans, we care about pigmentation because it's what protects us from the sun, from UV radiation. So we have a wide range of skin tones that people come in, and it generally depends on where your ancestors might have originated from. So the melanin that we create in our skin, it's the only type of pigment that we make, that helps protect against UV radiation from the sun, so it doesn't damage our DNA. And so people that have ancestors that have lived in really sunny areas near the equator tend to have darker skin and more melanin production naturally, versus people with ancestors that lived up in the North or South poles where there's not as much UV radiation to protect from. So just as a PSA, regardless, it's really important to wear sunscreen no matter your skin tone, so that you can prevent moles and things like this from becoming cancerous. But yeah, so we also have color patterns. It's just maybe not as bright and fun as the fish that I'm talking about. So when we're talking about fish color patterns, we can kind of classify them into different groups. We have uniform patterns like the fish in the center, which is just a plain orange color, right? There's not really any kind of special thing about the pattern. It's just one continuous color. And then we have fish like this box fish on the side here that have these spots throughout. And then we also have these different types of stripes. And the way that we differentiate between the two is that horizontal stripes going side to side, those are considered stripes, like on our zebrafish here down at the bottom. And then we have vertical bars. So we call those bars, like on the clownfish on the left. But typically what you'll see is that fish will have more than one type of these patterns. They won't necessarily just be one type of pattern. So you see stripes and bars and spots all over this fish. And this is a mandarin fish. It's native to Australia, the oceans around Australia, and it is also poisonous. So the blue should clue you in on that. So how did these fish form these patterns? So all bodies, not just fish bodies, are formed from different types of cells. And there's different types of cells that do all kinds of different things within the body. So heart cells to pump blood, immune cells, brain cells, blood cells, liver cells. And we even have specialized cells that their whole job is to create pigments. And in fish, we call them chromatophores. So chroma meaning color. So these chromatophores their whole job is to create pigment molecules or structures. And we classify them by the type of pigment or the color that they make. And over on the left, you can see that this is a zoomed in picture of the squid skin, kind of similar to what we saw with the GIF, the one that's changing. So you can see how they're kind of all arranged in these circular patterns. So you can see how the chromatophores are arranged in the skin. So they're right on the surface. Okay, so a bunch of different pigment cell types. We have melanophores in fish. Those create melanin. Okay, so they kind of look like this. And over here, if you look at fish skin up close under the microscope, you can see that the melanophores have a really dark pigment. They're pretty obvious. But one cool thing about them is that they can spread out or shrink down all of their pigment. So they can either keep it all really close to the center of the cell, or they can spread it out throughout the whole cell. So these have the melanin kind of spread out. Okay. And it's really similar to the same cells that we have in mammals that make, the brown color that we have in our skin and hair. And then xanthophores. So fish have multiple types, unlike us. So xanthophores is the next type. And those make carotenoids. So there's different types, but basically it's your oranges and your yellows. So over in our fish skin picture here, you can see the yellow orangey spots. Those are the xanthophores, like really up close. And then the iridophores are a little bit different because they don't create like a pigment molecule that just sits in the cell. They create these, they're called guanine plates, but basically they're structures that act like mirrors. And so they reflect the light that comes into the cell and reflects it back off. And that's what makes fish skin so shiny and iridescent. So if you've ever seen like fish scales up close and you can see like the shininess to it and like the kind of blue tint, that's because of the iridophores. So when you look at it over in the skin close up, you can't really see the outlines of them like you can the melanophores or the xanthophores. You can just kind of see the glint of shininess. So they're a little harder to study. But I do want to mention that all of these cell types are pretty small. So I think you guys had a talk with Dr. Z about nano stuff and what nano means. So maybe you remember how large she said human hair normally is, but it's normally about 60,000 nanometers wide. Right? Melanophores are 300 times smaller than that. So you can see the scale. This is 200 nanometers right here. And a lot of these cells are even smaller than that. So really, really tiny. And that's why we need microscopes to see all the stuff up close. And so all those different pigment cell types, they will move through a developing fish embryo. They begin their journey near the back, right along the spinal cord, and you can see them move and migrate through the skin. And this movement happens in all species, not just fish. Another example would be tuxedo cats. If anyone's ever owned a tuxedo cat or seen a tuxedo cat, you'll notice that they look like they're wearing a really dapper little suit. And that's because basically during development, all of their black melanocytes were moving from the spine, from their back, all the way to their front side and all the way down to the ends of their paws. And sometimes they don't have enough pigment cells to make it all the way to the end there. So wherever they just run out of cells, that skin and hair just becomes white. So every cat is different because every cat runs out of pigment cells at a different spot, but you'll tend to see the white being on the paws or on the front of the body because that's the furthest away from the spine, right? But this migration that we see happens in fish as well. This is just a really good, like common example. A lot of people know about cats. So cell interactions. These cells will move through the embryo, and then once they make it to the skin, which is their final destination, they'll start to interact with each other. So they will maybe repel each other away, or they might attract each other closer, or change the amount of pigment molecules that their neighboring cell makes. So they can communicate with their nearby cells and say, hey, you need to make more melanin or more carotenoids or whatever pigment, or you need to make less of it. And these interactions that they undergo change the whole overall color pattern. So you can see over on the left, this little video of a xanthophore, which remember is yellow, chasing a melanophore around a dish, so under a microscope. And it's just pushing it away. And when we look at it, or when we look at fish skin, we can kind of see the results of that. So this fish on the left, this is the zebrafish that I mentioned earlier because of the stripes. And when you zoom in on their skin, you can see that their chromatophores are arranged in kind of like these alternating striped patterns. So this interaction over here, that's literally what's happening in the skin. The xanthophores kind of arrange themselves together and then push all of the melanophores away. They just repel them in opposite directions. And so then you get these alternating blue and yellow stripes. In a really closely related species that does not have the same pattern, this is just more of like a uniform pattern, you can see that all of the pigment cells are just kind of spread throughout, just kind of randomly, right? They're not arranged in stripes. So this same interaction that's happening over here is not happening in this species. So the same repelling action isn't happening. So how do scientists actually study like the formation of these patterns in fish? So I'm a developmental biologist, and I wanted to give you guys another kind of challenge. Developmental biologists use a lot of different types of model species to study whatever we study, in my case, pigmentation development. But embryos in general are all really similar. So can you guys figure out which one is human? So 1 through 5, you can type it in the chat, whatever you think, whichever one you think is the human one. Yeah, so we're getting quite a mix so far. So I got a 1, a 2, and a 3. So I'll go ahead and reveal the results. Ooh, maybe. So actually number 4 is the human embryo. 1 is the zebrafish, we have a chicken, we have a 4-striped skink, and then we have a dog on the very end, everyone's favorite. So you can kind of see there's a lot of similarities between the embryos. We got a little head, and then it leads to a tail, head, tail, head, tail, head, tail. So early in development, any animal that has a spine or a backbone is really, really similar. And so even some of the other grad students in my department had a hard time being able to do this. So it's one of the nice things. We can use fish, for example, and study genetics and development in them, and then apply that to human embryos because they're so similar. So if one gene is working in fish, then there's a good chance it's working in humans as well. Okay, and so to show you guys what it actually looks like when we get a fish under the microscope to watch their development, I have a little video. So this is showing the first 3 days of development. You can see all the cells start to divide. So the sperm comes to the egg, fertilization happens, and then this timer right here is counting the number of hours after that. So I have a little diagram over to the side so you can watch as well. But the nice thing about fish embryos that we really like is that they're transparent. And also that they are fertilized outside of the body. So we don't have to worry, like with chickens, right? If you're trying to look at the developing chicken, you have to somehow see through the shell. Or if you're looking at a mouse, it's still developing inside its mother. Fish develop outside of either of their parents entirely. So they're really, really convenient. And so now you can see the head region starting to form here, and then the tail, and then the spinal cord and the bones start to come around, and the tail is going to kind of push out. And then by the time we get to about hour 26 to 28, pigmentation starts to show up. So the first cells to show up are the melanophores that create the melanin. So you can see them showing up now. And then they start to move through the embryo, and they'll arrange themselves mostly around here. It's also really dark in this region because this is the developing eye. You see a lot of pigmentation in fish eyes as well. So there's a whole field just dedicated to fish eye pigmentation. You see the little fins. The yolk is very small now. Soon, once we get to 3 days old, they'll start to start moving around. But you can see that now all of these melanophores have moved and they're all down here. They're not so much in the middle of the embryo anymore. So there they go, swimming off. So that's what we see when we look at them under the microscope. But what can we study about them other than just, watching them through a microscope? What else can we do? So one of the things that I use and a lot of other scientists use is gene editing. And to give you guys kind of like a crash course back into genetics, if you're unfamiliar, basically DNA is like the instruction manual that tells our bodies how to be built. Okay, so if we have that instruction manual, we might have one word in that instruction manual that tells you the end result. And that one word would be like a gene. So a gene encodes for a single type of protein, which can directly cause some sort of trait. So we could have a gene that encodes for a protein that makes eyes brown, for example. But if we change that gene, the eye color gene, and mutate it, then maybe suddenly we don't have brown eyes, we have blue eyes. And that's what happened naturally a long, long time ago. That's why some people have blue eyes or green eyes. It's just a mutation. But scientists can do this process a lot faster. So we can use tools to specifically change someone's DNA. And one application for this is to make a transgenic line. So trans means across, genic means genes, and to create a transgenic line means moving genes across from one species to another. So if you've ever seen the glowfish, like at the pet store, for example, the ones that glow really cool colors when you put the black light over their tank, then you've seen a transgenic animal. Because basically they were created from regular old zebrafish, like we've been seeing. And scientists took a gene from jellyfish that glow green, or lots of other different genes that glow different colors, and inserted it into our zebrafish. And that made them also glow green, just like the jellyfish. And so scientists realized that we could use this technology to help us as well. So other than, rather than just making the whole fish turn green, what if we just turned one part of the fish green, something that we wanted to identify or be able to label with like a color, right? It's like highlighting something in your notes. And so you can do that with like parts within a cell, like if you want to label a protein. You can also do it with a particular cell type, which is what I'll be doing. But how do we actually move a gene from one species to another? Like, how do we get a gene from jellyfish and move it over to fish to do this? So this is where it gets maybe a little bit more complicated, but the gist of it is that we use a method called Toll-2 transposase. There's lots of different methods to do this sort of thing. This is a little bit older one. It just happens to be what I'm doing. But you take the piece of DNA, whatever you might want to insert, maybe that gene from the jellyfish, for example, and you put it into this circular piece of DNA called a plasmid. So the plasmid is just like a little carrying purse for the piece of DNA that you want. And then you'll take your little purse with your DNA in it, and you will inject it into an embryo. So through a really tiny needle over here. And then with the help of this other enzyme called transposase, this little piece of DNA that you put here will be moved into the genome. So it's basically like taking a page that you've just written up yourself and stapling it into your instruction manual. But this happens randomly. So that little piece can go anywhere in the genome. It could go in chapter 1 or it could go in chapter 20. There's not really a way to control it directly. So to combat that, we have to inject a lot of embryos until we get exactly what we want, which is why I'm still working on this. But to get into what I'm actually doing, I'm working with this species, which is called Neolamprologus burchardi. It's the fairy cichlid, and it's a freshwater fish that's native to Africa, specifically like Tanganyika. It's the only place it's found in the world. And its facial pattern is really cool because it involves the 3 different pigment cell types kind of localized in their own little areas. So that lets us kind of look at just one cell type at a time, which is really useful for like comparison. So what I'm doing using the model species that my lab uses, I'm building a transgenic line of fairy cichlids. So the idea is that we could get these iridophores, the ones that are the really shiny cells, to get them to glow red during development. And this would allow scientists like myself to track iridophores through the embryo, because remember when we saw in that video, they don't have any color. So they're all clear really early in development. So if we can label them earlier than that, we can watch as they move throughout the embryo. We could also look at them in adult fish. And remember, those iridophores were really hard to see the borders of. But having some sort of label on there would let us have a good way to count the number that we see. So basically, this is building a tool that the lab would use in the future. So the first step is to make that plasmid, that little carrying purse for the DNA that I said before. And the idea is that we would use this gene called mCherry, which instead of making a green protein, basically it just makes it red, but it's a similar idea. And we take that gene and place it into our little plasmid carrying pouch. And then we would use that to inject our embryos. And actually the work to do this is kind of repetitive, and it's not super exciting. So it's a lot of sitting here at my lab bench and moving liquids into really tiny little tubes with a tiny little pipette. So there's not a lot to share with you guys about the process of that, but that's how we actually create the plasmid that we need to do the more interesting stuff. So then the next step would be to collect embryos from our little fishies. I don't have a video of our cichlids specifically because we have them in a secure room. So I'm not allowed to take certain like videos and things like that in there for safety reasons. But I do have online videos of zebrafish, which are similar enough. So basically how the process works when we want to collect all the embryos from them is we take a male and a female, place them into a tank together with a little divider down the center, like you see up here. we'll prop one side of it up a little bit. They actually really like it when there's shallow water. That's where they tend to mate out in the wild. And it's similar with our cichlids as well. So we prop it up a little bit. And then we will remove the divider. Keep in mind that we do all of this as soon as first light comes up because that's when they tend to mate. So it has to be right at the beginning of the day. Luckily, in our fish room, we have automated lights. There's no windows. So we just can artificially turn on the lights at like 8:30 a.m. So I don't actually have to come in at like 7 in the morning to get any embryos. But this is what it will look like when you actually remove that divider. So this is normal mating behavior. The male will chase the female around. And then eventually the female will release eggs out into the water and the male will release sperm. And that should fertilize the eggs. And then those fertilized embryos will, oh, there they are, there's the eggs. Those fertilized embryos fall down between this grate that we put in place so that the fish don't go down and eat their own eggs, which is actually a problem. So then after we have plenty of embryos, we can move on to the next step, which is prepping them. So we soak them in a little bit of really diluted bleach solution so we kill any algae or anything that's contamination. And then we make this gel mold over here on the right. So it's actually really similar consistency to Jell-O. And it's pretty much the same idea where you put your little powder into your buffer and you boil it and then you pour it and let it cool. It's a little bit firmer and it's not nearly as tasty. But with the little mold that we have made, we have these grooves and the little round embryos fit in those perfectly. And that basically means that the embryos can't move around as we poke them with the needle, which is in the next step. So that helps keep them in place. And then here's what they look like under the microscope with our microinjection needle. We also use a dye so that we can see which ones we've injected. So we have the gene editing materials and the dye. And then we use this little foot pedal thing that pushes really tiny, tiny amounts of our material into these embryos. So you can see this one's already been injected and then this one's just poked through. And then after that's been done, after you do rows and rows of those, you can look at them under the same microscope. And this little box on the right makes the fluorescence work. So it puts off certain wavelengths that make the embryos fluoresce if they've been correctly done. And then at the top, we have a camera so that we can see everything that we're doing. And all of that stuff can show up on the monitor over on the right. So this is my little setup. And then since I haven't actually been able to get one labeled exactly the way I want, I'm just going to show you guys an example of what one successfully transformed looks like. But these are all zebrafish that have been had GFP, that green one from the jellyfish inserted into their genome. And remember I said it was a really random process. So each one of them had the GFP placed in a different area. So like in a different chapter of the instruction manual. So if they had it placed in like the nervous system chapter, then they're probably going to have the nervous system structures glow green, so on and so forth. So this is basically what it would look like. And that would help us see if there are any abnormalities with certain embryos with our pigmentation in the future. So we basically get to watch all of our little pigment cells move around, and it'll be a really, really cool time once it actually happens. So, okay, I think with that, it's everything I have for you guys. Hopefully it wasn't too fast. Tried to fit in a lot of stuff, I think. Are there any questions?
I have a few, but I'll wait for the other students. This was
Yeah.
amazing. I was actually so intrigued. I was staring at it,
Oh,
right?
I was so worried because it's so different giving a talk to people that I can't really see. So I'm like,
Right.
am I boring everyone to tears right now? I
Oh
have no
no, at
idea.
least not me, because I was like, wow, I didn't even know this, right? But, okay, so if anybody else has a question, you can come after me. There we go. There's Isabel.
Okay. Oh, good. Okay. Thank you. I'm glad you guys liked it.
Oh yeah, it was really good. But are there any types of fish where their pigmentation develop over time, like as in years or even centuries?
I'm not sure about the centuries one, because I mostly work with like a smaller freshwater fish.
Mhm.
So usually the ones that grow really large like that and live for a long time tend to be ocean fish, or like really deep water fish. And their pigmentation tends to be actually pretty dull, because no one's really going to see it anyway when it gets that dark. So I'm not sure. I'd have to look into that. I imagine it could change over time depending on like social, uh, like where they are in the hierarchy and things like that, but I can't think of like a specific species.
Great, thank you.
Mhm.
There was, oh, I didn't write this one down, but it's in my head still. But have you like you see the one that you have pictured there? It's pretty like just one color, right?
Mhm.
Have you seen them change from like a single color to multiple colors?
Oh, like go from like a uniform color to like different patterns.
Right.
Sometimes that can happen. Uh, so I guess this kind of goes along with your last question. Sometimes that can happen like during development. So if the fish go through like the different life stages, just like people do, and they have like a little teenager stage, the juvenile stage. So sometimes the juvenile fish of a species can look pretty different from the adult. So sometimes they can completely change their pattern and their whole body pattern, like just their size, and like their shape in general. So sometimes it can change a lot. Um, for a specific example, I don't know if I have one at the moment, but but I know that that happens pretty frequently.
Okay. You gotta be wondering now after I.
Yeah, and I have to look up examples because I'm sure they're out there. I just cannot think of one at the moment.
Yeah, I saw Nemo and then I was all like, oh, but
Yeah. Well,
I,
yeah, well, they don't even just change their pattern, they change their sex. So they go back and forth between male and female.
oh wow, did not know that.
Yeah.
Very interesting. Anybody else have any? I have another.
Do you have a favorite project you have ever worked on, favorite class you took or are taking in college that you would recommend to those who want to go into a similar genetics field? Um, oh, the most interesting fish I've ever learned about. Okay, so favorite project. Um, a favorite project. I don't know. I think the cloning stuff that I'm doing now is pretty cool. Mostly because I just like anything that has microscopes involved. So even when I was at Mayo Clinic, the stuff that I was doing, I used a little bit of microscopy, which I really liked. So that's one of the things I really liked about coming to ISU and doing the project that I am now. As for my favorite class I took in college, actually might be surprising because it has nothing to do with fish, uh, or like anything that I'm looking at now. But my favorite was probably nanomedicine, which was taught by a visiting professor from Mayo. So they came over to our university and taught us about it. Nanomedicine is really cool because it's basically like taking drugs, so like cancer drugs and like things for treatment, and then loading them into like these little vesicles, these little things that they can use to move the drug throughout the body, which is really useful because it helps it like hide it from the immune system, and it helps it from like building up in the liver or getting like flushed out, because most cancer drugs don't actually make it to the tumor. So that's something I learned there that I thought was really, really neat. I did apply for that PI's lab while I was at Mayo, but yeah, her class is really cool. And she has a TED Talk on YouTube if anyone's interested. Her name is Joy Wolfram. So she gives a little talk about nanomedicine on there, which is awesome. The most interesting fish that I've learned about, oh gosh, there's so many. Have you guys ever seen a hagfish by any chance? I don't know. I could potentially find like a video. Let me see. Just to show you guys what I'm talking about. Hagfish are really cool looking. So yeah, so let's see. Okay, so just a quick Google. Hagfish are really interesting because they create this like slime as a defense mechanism. You can see here. So it's like, it's kind of looks like an eel. So just like a little cylindrical body, but if it feels threatened, it creates this like super viscous slime, which can just like coat everything. I thought that was like the weirdest thing I'd ever heard of when I learned about them, but also cool. I kind of want to hold one just so that I can feel the slime because I'm really interested to see what it feels like. But hopefully it's not too gross. I think that's really cool though. And then for ornamental fish, do you think special diets such as one with pigment enriched diet will make the coloration better, or is it purely genetic? So I think that stuff can help in certain cases because obviously to make those pigment molecules, it's you need all of the ingredients to like mix that stuff up, right? So if they're not getting enough of that stuff in their diet like they would naturally, then they're going to be more dull. We don't use that type of enrichment for our fish, but I know that it certainly can work in some instances because obviously you're taking them out of their normal natural environment. You're not necessarily feeding them all the same things that they would. So they're not getting all the like sorts of chemicals and nutrients that they would need to make those things. Okay, courses or classes you recommend? Definitely, I mean, genetics is a given. If your university offers like a developmental biology, or sometimes it's called embryology class, really, really helpful. I'm trying to think. Sometimes you have like these specialty classes that just pop up out of nowhere in college, and like a professor is just teaching them for a semester because they're really interested in the topic. And I would definitely suggest trying out at least one of those courses while you're in college because it might surprise you how much you like it, because that's what happened with nanomedicine with me. Anything that you think sounds interesting, just take it, because you never know when that information is going to be useful, right? And it might help you discover a passion for something that you really want to study in the future. Yeah, I don't know if you're talking about like for the curly hair naturally, like putting your hair in your product or putting product in your hair. You mean for the pigmentation? Yeah. So for the fish, I think it still works in that case. Yeah. There's a lot of like supplements for humans that I would say would not work to help like enhance things. But in the case of fish for pigmentation, I think some of those things can be useful. I'm just not super knowledgeable about astaxanthin, astaxanthin, probably xanthan. We have any other questions?
I do. I have one. Has anything human triggered, such as climate change, have any effect on the pigmentation? Have you guys noticed?
Um, with the species that we're looking at, that our lab mostly uses? No, not so much. But I'm not really involved with going out and collecting them. My professor is actually the one that takes those trips. So if anything, he would know the most about that. But as far as I know, I don't think that we've seen anything like that. The bigger issue has been more with invasive species, especially in Africa. So there's a lot of issues with people releasing animals that shouldn't be released. And so it's eating a lot of these fish that are only found in one lake in the whole world. And anytime you decrease the number of a population by a lot, then you're going to see a lot less diversity or genetic diversity, which could eventually impact pigmentation.
Oh, thank you.
Recommend taking internships and summer programs. Yeah, I think they're really helpful. I wish I kind of would have gotten into it in high school if I had had the opportunity, but I just didn't even think about it as an option at the time. I'm the first person in my family to be a scientist. My parents both went to college, and my dad even got a master's degree, so I'm not unfamiliar with that stuff, but I just didn't realize that with science that it was so important to get, I guess, experience as early as possible. And another thing that if you guys do end up wanting to go into research, I can't suggest more, that I definitely messed up when I applied to grad school, is to directly message professors. Tell them that you're interested in their research and that you'd like to come in and, shadow someone in their lab, or if you'd like to intern, depending on where you're at in your schooling. And generally, I found that professors can be really, really supportive. They want to bring people into science. So don't be shy to kind of reach out to people. And if, an email doesn't, or if they don't send an email back right away, sometimes they're just really busy and they're not trying to ignore you. So, send another one. And if that doesn't work, then contact someone else. But yeah, I would suggest doing it if you have the opportunity, for sure.
Now you see why we schedule these so far in advance, right? Because
Yeah.
everybody's so busy. You have to get a 2-3 month jump.
Graduate students run into it too, because we need the professors to be on our graduate committees and things, and help us out with that stuff. So you have to schedule committee meetings, or any of the big test things that you need your committee for. You have to schedule that stuff months in advance, because you got to get 5 professors or 3 professors in the master's case all together. So at the same time, it's a nightmare.
Yeah.
It's literally a nightmare.
I think I when I was doing my thesis, I was crying because I had to start all over after 8 months, right?
Oh my God. My boyfriend is a PhD student in the same program, and he actually just defended his PhD. And he was really close to missing the deadline for being able to do so, because we just couldn't find a time for all the people on his committee to be there at the same time.
Oh yeah, it happens.
Crazy.
What's your favorite process during your project?
You mean with what I do in the lab, or?
Yeah. Mm-hmm.
I think this is a little tiny thing. I work with bacteria as well, so they help me out with the cloning. The plasmids that I was talking about, that are the little carrying pouches for the DNA. We put those inside of E. coli bacteria, and then grow up lots of copies of the bacteria. And so they make lots and lots of copies of that gene. And one of my favorite things is when you have a plate of bacteria, and then you take a little tip thing and pick just one colony off the plate. I don't know, it's super satisfying. That in the microscope stuff, because it's always fun to see stuff under the microscope. I feel no matter what age or what skill level you are as a scientist, it's always going to be fun to see stuff under the microscope.
Yeah, I agree. All right. we're coming at the bottom of the hour. So anybody have any more questions for Peyton? And Peyton, if they have questions that they forgot or it didn't come to mind while we were here, is it okay for me to email you any more questions?
Yeah, of course. Yeah, I'd love to answer. And then I might be able to actually do a little bit of research and show you guys some examples.
Oh, that'd be great. Let me turn off the recording real quick so we can